Hafaday, I'm Allie, and this is a video to explain to my mom what it is that I do for work as a marine biologist out in the field. So I'm currently on NOAA's ship, the Reuben Lasker, and we are on a Coastal Pelagic Species Survey. This is like 3 of the survey, going from San Francisco, California to Newport Beach, Oregon. Now you might be wondering, what are coastal pelagic species? And why are we surveying them? Let's ask our chief scientist. Okay, I'm with Owyn Zach. He is the fisheries research biologist. So what are coastal pelagic species?
Coastal pelagic species are an assemblage of fish that range from sardine to jack mackerel, Pacific herring, and anchovy. And they basically make up a lot of the forage that you find off California, the forage fish, essentially. Pacific's, market squid is also part of the assemblage, but these are what we're targeting right now for this survey. And they're a fish that generally lives in the open ocean, anywhere from a couple miles off the beach to a couple 100 miles off the beach.
Sweet. So if you live in California and you love your seafood, chances are you've probably eaten the coastal pelagic species that we're surveying. They're lower on the food chain, and their prey include zooplankton and phytoplankton. But their predators include yellowtail, tuna, and even whales. Now, the reason why that's important is because if we were to remove the coastal pelagic species from the food chain, these larger pelagic species, the whales that are migrating through California's coast right now to feed on the abundant sources of pelagic species that we have out here, those whales would suffer and they wouldn't migrate out here if it weren't for these species that we're surveying. So let's get back to Owyn to learn a bit more about why it is that we're here on this survey.
We're trying to quantify the biomass of Pacific mackerel and sardines and anchovies in the California current, so we can collect data on how many fish are out there, and then that gets fed into stock assessments and then fisheries management.
Okay, thank you, Owyn.
No problem.
We're figuring out the abundance, the distribution, the quantity of coastal pelagic species across the West Coast region, from Mexico to Canada. But now to the next question. How do we do that? Let's head to our acoustics lab to find out. All right, so I'm here with Dr. Allison White. She is a specialist in acoustics monitoring.
Sure.
And what is it that we're using hydroacoustics for in this lab?
Sure. So we are utilizing what's called active acoustics or hydroacoustics, which is where we send sound into the water and we measure the intensity of sound that gets backscattered or reflected off of objects.
So we're taking a look at one of these graphs right now.
Yes, so we visualize the sound in a 2D display that's called an echogram. So here we have, this is the strongest return we get, which is from the seafloor. And then this is right underneath the ship, so roughly the surface of the water. And then this is all of the stuff that's between the ship and the seafloor right now. So these blues and greens are sort of the weaker intensity responses we get. Mostly these come from things like zooplankton or suspended sediment is usually blue. Sometimes you'll see these like kind of more greens with some yellows. That's more like what zooplankton look like at this frequency. This is collected at 38 kilohertz or 38,000 pings per second. And as we scroll through, so this is time or distance that the ship traveled. We get to see, this is some rocky bottom habitat with, these are some fish schools on top of it. But we're really targeting in this survey are coastal pelagic species, which generally look something more like this. So these really dark reds are stronger intensities that we get off of things, especially that have any gas in their bodies like most coastal pelagic species do.
We use hydroacoustic monitoring data to locate our coastal pelagic species. And we do that by covering 3 different distances during the day with our vessel. You can see these 3 distances or transects going from inshore to farther offshore. And within each of these transects, we figure out exactly where the density of our coastal pelagic species may be the highest using hydroacoustic monitoring data. At night, we visit each of these sites and we conduct our trawls.
Swoosh! All right, now it's voiceover time. I'm currently at the bridge of the ship, the area where we have our ship's control board, and I'm about to conduct marine mammal watch. So we do this before every single one of our trawls, and it's to ensure that there are no marine mammals in the area that we are going to trawl. We don't want them to get caught up in our net, and we also don't want to disturb their feeding grounds. So I'm navigating out to the flying bridge of the vessel with a red light. We need to make sure that we're maintaining the crew and myself's night vision. And once we get out there, I am on the lookout for any whales, sea lions, dolphins, listening for the blowhole of a whale, the chatter of a sea lion. And wow, what a bright night. I promise it's better with a human eye when you're out looking at the water, but when there's no full moon, it can be a bit difficult to see. If we spot a marine mammal, we move a nautical mile away from them, but if not, we plug in our data and we are good to go. We also have a camera that we use to attach to the net to see things that we otherwise wouldn't be able to see. We want to make sure that the net is functioning properly, and if there are any other animals like sharks or larger fish, they are able to escape through the openings in the net, and we can check that with the camera. After that, we have what are called TDRs. Now, TDRs are time depth recorders, and you can see Owyn plugging in the TDRs in order to get our data. We also attach these to the net, and once they're attached to the net, as I had mentioned before, they are measuring the time that the net hits the water and the depth of our net to make sure that we are at the depth that we want the net to trawl at. We then have the crew place the camera on the net, we grab our TDRs and put them on there, and then it's time to go fishing.
All right, it is 4 a.m. and this is our 3rd and final trawl of the night. I'm currently in the wet lab, but we are walking out to go see them bring the net in. So the net is placed around 0 to 35 meters from the surface of the water, and it can hold up to 8,300 pounds, which is 2.5 Toyota Camrys, the car that I drive. We're bringing the net in right now, and we'll go ahead and see the haul that we have soon.
All right, so after all of that prep work, here is our haul from half an hour of trawling. We stick all of that into these giant bins, and from these bins we have a bunch of jack mackerel, which we want to put into these orange buckets you see. We record the weights of each of our orange buckets, and here you can see us doing some line work. One of us puts all of the contents of the white bins into an orange bucket, then 2 people take that, carry the bucket over to a scale to measure it, and we have 1 person squeegeeing to make sure we don't trip on any foreign jellies or things that might be on the ground. There's our beautiful jack mackerel. Delicious. And after that, we take the measurements of each one, their lengths, their weights, and all of this data is going to be recorded and sent to the NOAA Fisheries Science Center. They're going to process all of the data, and from that, we'll learn about the abundance of our species, the distribution of our species, and their biomass. Look at the teamwork, the collaboration, innovation that excites. We love NOAA Fisheries, exciting work. What else do we do with our fish? That's right, it's dissection time. So we're looking for the otoliths within our jack mackerel, and that is their inner ear bone structure. Their inner ear bone, or the otoliths, help them to hear, sense vibrations in the water, and maintain balance, which is all very similar to the way our inner ear works. Now, I'm cutting through the
back part of the jack mackerel's head, and that's because otoliths are located directly behind the brain of a bony fish. Now, most fish lay down a distinct bone structure by the season in the winter and the summer. They're so distinct, they're like tree rings. In the way that we can count tree rings and age a tree, we're able to count the rings on an otolith and age the fish. These otoliths are also used for isotope analysis, which helps us understand the water source or location of the population, and biochronology, so we can understand the rate at which a population is growing. Here I am taking out some of that brain. Yum. Girl dinner. Putting it down. You can see what the top of its head looks like. And alright, it's time to speed this up. Do 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 do.
Was mine 7?
Playing hide
Yep,
and go
yours
seek.
was lucky number 7. You got him?
I think I got it.
Nice.
Yay!
Correspond to
Yes!
3 axes
An
in
otolith!
the water column.
Yes!
So pitch.
Now, to keep track of our otolith samples, we put a barcode on each one of the samples, and these end up getting sent over to our Fisheries Science Center for processing. Woohoo! Time to barcode all of them. There's a lot. There we go. And once we do that, we scan them. Scanning them will put them into the system, and all of the data will be uploaded for each of the otoliths. Now you know what time it is? Trawl time again. That was round 1. We do it 3 times. Here we go. Same situation. Put them in buckets. Once we put them in buckets, we take 5 buckets for our sample size. We throw them onto the table and we sort through them. What we're looking for is one of each species that we come across, as well as our coastal pelagic species. We collect one of each species so that we can understand the diversity of the samples that we're collecting, and then we want to make sure we're finding our coastal pelagics. Boom, some smelts, super organized board. That's typically what we do, lay them all out in an orderly fashion. Here's an orderly fashion. That's Pacific hake, and these are going to be used for the Northwest region. And sometimes we get a bunch of jellies. If you like SpongeBob, you might recognize these ones. Moon jelly. Aquaria. Egg yolk.
Do not touch it.
Lion's mane.
Okay.
Sometimes we come across species that are more rare than others. This one is a robust clubhook squid, and it is the 2nd largest in the North Pacific Ocean, growing to be up to 12 feet long. We're donating this one to Scripps Institute of Oceanography's Pelagic Invertebrate Collection, and here you can see a ton of different samples that we take in the duration of our survey to send to other organizations, universities, and agencies to help with their research. And sometimes we come across my favorite fish in the world, a mola. That's a sunfish. They can grow to be up to 14 feet long. They eat jellies for breakfast, lunch, and dinner. They have that cute little beak. And I've been told that they look like they stopped in the middle of evolving, but don't listen to the haters. Also, thank you so much to Owyn and Zach for showing us the ropes and answering the many questions that I had to ask the both of you throughout the survey, as well as Allison for her guest appearance. And thank you to the crew for supporting everyone on the vessel, our operations, and making people that are new to the team and here for a short time feel very welcome. I really enjoyed my experience here, and it's because of the warmth of everyone on the Reuben Lasker. So, huge shout out Reuben Lasker. Let's go team. I love the Reuben Lasker. And that wraps up everything there is to know about our Coastal Pelagic Species Survey. So thanks for listening. If you have any questions, feel free to ask me. And if you're not my mom, get fishing. Happy birthday to you. Happy birthday to you. Happy birthday, Mom! I made this video because you've always supported me through my endeavors in marine science, and you've been the number one person that asks me all the questions there are to ask about the type of work that I do. So this video is for you, Mom, so we don't have to text back and forth about what a coastal pelagic species is. I'm sorry that I can't celebrate with you in person, but I promise we will when I'm back. And I just wanted to say thank you for pushing me when I haven't been sure of my abilities, for showing me that the sky is a limit, for giving me the spirit to explore, and for teaching me how to be the best person that I can be. I love you.